Welcome to the Department of Genetic Engineering, SRMIST, Chennai. This is Dr. S. Iyappan, Associate Professor, Department of Genetic Engineering, SRMIST. Today, I am going to explain light reactions of photosynthesis. Photosynthesis is the process by which green plants, algae, and some bacteria use light energy from the sun to make food. They convert carbon dioxide and water into glucose and release oxygen as the byproduct. That's what the oxygen we breathe. This process takes place in thylakoid membrane of the chloroplast. Photosynthesis happens in two main stages: the light-dependent reaction, which occur in the thylakoid membrane, the light-independent reaction, also called as Calvin cycle, which occur in the stroma. Light reaction can be divided into photosystem 1 and photosystem 2. When the light hits photosystem 1, it excites the special chlorophyll molecule known as P₁₁₀, and the excited P₁₁₀ loses its electron. These electrons are transferred through chlorophyll molecule A0 and phyloquinone A1 to iron sulfur cluster. This iron sulfur cluster passes this electron to ferredoxin, a small iron containing protein. Finally, ferredoxin transfers this electron to NADP+, which further takes H+ ion and converts into NADPH by the enzyme called NADP+ reductase. This NADPH is then used in the Calvin cycle to reduce carbon dioxide into glucose. Photosystem 2 begins by absorbing light in a wavelength of less than 600 nanometers. This excites a chlorophyll P₆H₁₂, which then loses its electron. The released electrons are passed to a molecule called pheophytin, then plastoquinone, and eventually move through a sequence of electron carrier and finally reaching a electron deficient photosystem 1. Now, since P₆H₁₂ lost an electron, it needs electron back to become neutral molecule. That's where splitting or photolysis come in. An enzyme complex called oxygen evolving complex in photosystem 2 split water into oxygen, protons, and electrons. This electron goes to P₆H₁₂, and the protons stay in the thylakoid space, helping to create proton gradient. What happens during this electron movement? Photosystem 2, light hits P₆H₁₂, and the electron goes from P₆H₁₂ through a sequence of carrier such as plastoquinone, and then transferring H+ ions to the thylakoid lumen and recycling the plastoquinone. From plastoquinone, this electron moves the cytochrome Bf complex and iron sulfur cluster to plastocyanin. Plastocyanin passes this electron to photosystem 1. Again, when the light hits on photosystem 1, the electron moves from P₁₁₀ to ferredoxin through a sequence of electron carriers. Ferredoxin gives this electron to NADP+ reductase, which makes NADPH from NADP+. Along with the NADPH production, the light reaction also produces ATP. As an electron moves through an electron transport chain, protons are pumped into the thylakoid lumen. This creates a proton gradient, lots of H+ ion in the thylakoid and fewer in the stroma. To balance this, protons naturally want to flow back to the stroma, and they do this through an enzyme called ATP synthase. As the proton flow through ATP synthase, it spins like a turbine and combines ADP and inorganic phosphate to form ATP with the help of ATP synthase. So The reactions of photosynthesis creates high energy compounds like ATP and NADPH, which are going to be used in the dark reaction to synthesize carbohydrates from carbon dioxide and water.